Hello my dear friends, greetings from Cubata Mercury and a warm welcome to the discussions part. I hope you will appear for an exam, the Cubata NEET model exam conducted today morning. Right. And right now, it's our motive to associate together to discuss those questions, from the same examination. And hereby we will touch all those 19 botany chapters, 11 from +1 syllabi and 8 from +2 syllabi. And I hope and I believe it will be a quick revision for you. So, be patient, be conscious, and be confident. Shall we move ahead? And now this is the discussion session meant for the botany. Okay? Here comes the very first question. Listen to the question. Ectocarpus is a. As you know, Ectocarpus is a brown alga, right? Ectocarpus that came together with the laminaria, sargassum, fucus, etc. Options are here like A. Filamentous algae, B. Unicellular algae, C. Giant algae, and D. Colonial algae. You know, the colonial algae that is familiar to you people is a volvox, and its colony is said to be the Cenobium. The colony is called the Cenobium. And that volvox, the Cenobium, the colonial algae, that is coming under the chlorophysi, the green algae. So Ectocarpus is not at all a colonial algae. Instead, volvox is the colonial algae, and its colony is called the Cenobium. Option C is here as giant algae. As you know, the giant algae members are said to be the kelps, the massive algal bodies, the marine massive algae said to be the kelps, for example, laminaria, sargassum, etc. But also we cannot find Ectocarpus. Unicellular algae. Truly, actually, there is nothing so. There is no unicellular algae at all. Actually, this unicellular algae, that means they are considering the Chlamydomonas and Chlorella. Truly, they are coming under kingdom Protista. They are not at all plants. They are not at all coming under the algae, as the Chlamydomonas and Chlorella are Protestant members. As they are unicellular eukaryotes, the Chlamydomonas and Chlorella were best placed under kingdom Protista. Based on the 5 kingdom classification proposed by the R.H. Whitaker, all the plant kingdom members are multicellular in nature. There is no unicellular algae at all actually. So, there is no scope while discussing this unicellular algae. And thus, the right answer meant for this first question is a filamentous algae. Ectocarpus is the filamentous alga with dichotomous branching, right? Dichotomous branching is shown by some of the brown algae members, and they are filamentous in nature. Okay, I hope it's okay for you. Let us see the second question, right? Shall we move on? Yes, here comes the second question. Listen to the question carefully. The main difference between gram-positive and gram-negative bacteria is gram-positive and gram-negative bacteria differs in which feature? Options are here like cell membrane, cell wall, ribosome, and mitochondria. As you know, the staining property of bacteria to classify them as gram-positive and gram-negative, that was described by a scientist called Christian Gram, and thereby it's said to be the Gram staining. The components of Gram staining are: the primary stain is the crystal violet, Its mordant is iodine. Its de-staining agent is alcohol, the ethanol, and the counter stain is the safranin. Yeah. And you just consider these options too. Regarding mitochondria, mitochondria totally absent in prokaryotes. Right. All these bacteria were included under kingdom Monera. The kingdom Monera is a prokaryotic kingdom. Thus, in both gram-positive and negative bacteria, the mitochondrion is totally absent. And regarding ribosome, the ribosomes are of 70S in all these prokaryotic members, where S is the sedimentation coefficient called the Swedberg unit, and the 70S ribosome is made of a smaller 30S subunit and a bigger 50S subunit. Together is the 70S ribosome. S is the sedimentation coefficient called the Swedberg unit. So, there is no difference in the ribosome of gram-positive and gram-negative bacteria. So, let us leave that option too. Regarding cell membrane is quite unique in all the organisms, and the basic structure of the membrane is quite normal in all the members, including prokaryotes and eukaryotes. That means the cell membrane is the lipid bilayer and impregnated with the proteins, either the peripheral proteins or the integral proteins. So there is no scope of discussing the cell membrane also regarding the gram-positive and negative members. And the right answer meant for this question is option B, that is cell wall. Due to the difference in the cell wall, the bacteria are of two types. They are the gram-positive and gram-negative bacteria. And you know, the staining is the property of the cell wall. In cell-walled organisms like plants, fungi, bacteria, etc., the stain is taken up by the cell wall itself. Due to the change in the cell wall, the bacteria are of two types like gram-positive and gram-negative described by Christian Graham. Okay, shall we move on to the third question? Here comes it. What is common for the structure of cell wall of bacteria and fungi? What is common for the structure of cell wall of bacteria and fungi? Options are here. Option A, both are made up of cellulose. Option B, both have mucopeptide. Option C, both contains N-acetyl glucosamine. And option D, both are made up of chitin. Let us see. The first member mentioned here is bacteria. Bacteria. As you know, in bacteria, the cell wall is made of peptidoglycan. Peptidoglycan. The cell wall is made of peptidoglycan. Right? Peptidoglycan. Or else is said to be the murine. Peptidoglycan or murine. We are talking about the eubacteria, right? But in archaebacteria, the cell wall is a pseudopeptidoglycan or a pseudomurine. And in the specific eubacterial members like cyanobacteria, the cell wall is made of peptidoglycan together with cellulose, right? So, anyway, in most of the eukaryotic bacterial members, the cell wall is made of peptidoglycan or murine. That peptidoglycan or murine is made of amino sugars, right? Amino sugars. The nitrogen-rich polysaccharides are said to be the amino sugars, right? The amino sugars are of two types in bacterial wall. They are N-acetyl glucosamine, the so-called NAG, and N-acetyl muramic acid, the so-called NAM. Both they are amino sugars, the nitrogen-rich polysaccharides, N-acetyl glucosamine and N-acetyl muramic acid. Okay, together with those amino sugars, the normal polysaccharides are also there in the peptidoglycan or murine. And regarding the fungi members, the cell wall is made of polysaccharides and chitin. Okay, the normal polysaccharides together with chitin. The chitin. What is chitin? what is the component of chitin? Chitin is again an amino sugar, right? Chitin is an amino sugar, right? Which amino sugar actually it is? That is a NAG itself, N-acetyl glucosamine, the NAG. So, what is common about the cell wall of bacteria and fungi? In both of them, the NAG is present. NAG is present in both bacterial wall as well the fungal wall. The cellulosic wall is usually shown by the plants, right? And the chitin wall is seen in a fungi. That chitin is an amino sugar called the NAG. So you may just look at the glance to the options. Both are made of cellulose? No, not at all. Both have mucopeptide? No, never. Both contain N-acetyl glucosamine? Yes, exactly. That is the right answer meant for this question. Both the bacterial wall as well the fungal wall is made of NAG. That is N-acetyl glucosamine. That is nitrogen-rich polysaccharide, otherwise said to be an amino sugar. In fungi, the chitin, the NAG, is otherwise said to be the fungal cellulose. That's the third question. I hope it's okay. Listen to the next question, question number 4. Here comes it. Yes. Which of the following is not characteristic of gram-positive bacteria? So, of these 4 options, 3 of them are the features of a unique group, and that is the gram-positive bacterial characters. But one alone is not a feature of gram-positive bacterium. Which one it is? Options are here. Cell wall is smooth. Option B, mesosomes are distinctive prominent. Okay. Option C, basal body of flagellum contains 2 rings, and murine content of cell wall is 70 to 80%. Of these 4 options, one alone is not a character of gram-positive bacteria. Which one it is? Yes, that's the question. Okay. So, cell wall is smooth. That is the feature of gram-positive bacterium. Mesosomes are distinctive or prominent. That again is a feature of gram-positive bacterium. Basal body of flagellum contains 2 rings. Fine. But the murine content of cell wall is 70 to 80% is not a feature of gram-positive bacterium. Instead, is a feature of a gram-negative one. Thus, the right answer meant for our present question, question number 4, is option D. The last option is the right answer. Okay. Let us move on to the next question, question number 5. Prions. Prions are agents of the following diseases. Yeah. The topic is coming under the chapter biological classification, and it's a newly added category under this chapter, prions. A. Creutzfeldt-Jakob disease, usually said to be CJD. CJD, Creutzfeldt-Jakob disease. Option B, bovine spongiform encephalopathy. The usual term is BSE. Bovine spongiform encephalopathy. Option C, Jessman-Stroessler-Scheinker syndrome. And all of these. Yes, you may just assess yourself and try to get the best and the true answer. Yes, all of these is the right answer. Prions, what are prions? They are infectious proteins. The size is equivalent to that of viruses. They are unusually folded proteins, infectious proteins without genetic material. And they will cause a neuron degeneration disease. That neuron degeneration disease is said to be bovine spongiform encephalopathy in medical science. The modern medical science is calling the neurological disease caused by prions as BSE, bovine spongiform encephalopathy. Its variant, its analog is said to be mad cow disease, right? Mad cow disease, right? The mad cow disease in cattle. The BSE is especially said to be mad cow disease in cattle. and its analog in humans can be known as Creutzfeldt-Jakob disease or Jessman-Stroessler-Scheinker syndrome. Yeah, these two things are the BSC in humans. So all these options are the diseases caused by the prions itself. Prions are unusually folded infectious proteins equivalent to the size of viruses, okay, without genetic material. That's it. Creutzfeldt-Jakob disease, the CJD, that's there in our NCERT. Bovine spongiform encephalopathy, BSE, that also is there in our NCERT, but it's a new term, right? Jessman-Stroessler-Scheinker syndrome, that itself is a Creutzfeldt-Jakob disease. All right, okay. Shall we move on to the next question? Okay, here comes it. Lichen is the pioneer vegetation on which succession? Chapter Ecosystem, 12th Botany chapter Ecosystem, and the topic Ecological Succession. Options are Hydrocere, Lithocere, Samocere, and Xerocere. Lichen is the pioneer vegetation on which succession? Here, two options are more or less okay. Okay? It's not Hydrocere because in Hydrocere, in hierarchical succession, that means the succession in wetter area, the pioneer species, the first species invade a bare region. The pioneer species in hierarchical succession or Hydrocere is a phytoplankton stage. Okay, and it's not Samocere too. Right, Samocere, the succession in sand, that also is not at all applicable here. But the Xerocere and the Lithocere. Xerocere means the succession begins in a dry area. Okay, the succession that starts in a dry area is said to be the Xerocere, and Lithocere means the succession that begins in rocks. Right, so these two options are more or less okay, but the best accurate one, the prior option is Lithocere. That means the succession on rocks. That's a kind of Xerocere. That is a type of hierarchical succession. Hierarchical succession means the succession that begins in a dry area. A special type of hierarchical succession, Xerocere, is Lithocere, where the succession begins on rocks. So the priority goes to Lithocere. If the Lithocere option is not there, if the Lithocere was not there, then you may just move for Xerocere, right? So the priority answer is Lithocere, and the second priority alone is Xerocere. That is the succession priority meant for lichens as the pioneer species. Okay, yes. Here comes our question number 7. Listen to the question number 7. Pyrenoids. Chapter Plant Kingdom, is it? Division algae, especially green algae, the chlorophysi. Pyrenoids are made up of. Options are here: core of starch surrounded by sheath of protein, core of protein surrounded by fatty sheath. Option C, proteinaceous center and the starchy sheath, core of nucleic acid surrounded by protein sheath. You know, under the plant kingdom, the first division is said to be algae. The oldest, simplest, the primitive aquatic plants are said to be algae with a thalloid plant body. They have chloroplast. Chloroplast is a double membrane bound cell organelle, right? A double membrane bound cell organelle is the chloroplast. Within the chloroplast, they will be having the reserve food. Is it? The reserve food is the starch, but the starch is coated over proteins. The central core is made of protein, and above that protein central core, the starchy sheath is coated. Starch is on outer side. The protein is at the center as the core, right? So, core of starch surrounded by sheath of protein? No, the case is just reverse in the pyrenoids. Core of protein, okay, this one is okay. Surrounded by fatty sheath? Not at all. Proteinaceous center, yeah, protein rich center, the core is made of protein, right? And starchy sheath around, exactly, that is fine. And core of nucleic acid? Nucleic acid means DNA or RNA, which is not at all, right? So not divided by protein, that also is not at all possible. So the right answer means for the question number 7 is option C. That means a proteinaceous central core with a starchy sheath around. That is said to be the pyrenoids. Pyrenoids are the reserve food material in chlorophyll, the green algae, and they are present within the chloroplast. I hope it's okay for you. Right. Listen to me. Here comes your question number 8. Listen to the question. If you are asked to classify the various algae into distinct groups like a chlorophyll, pheophyll, or rhodophyll, which of the following characters you should choose? Yes, a cunning question, right? Yeah. Types of pigments present in the cell. Option B, nature of stored food materials in the cell. Option C, structural organization of the thallus. And option D, chemical composition of cell wall. Yeah, under the chapter plant kingdom, there was a table likewise, the 3 different classes of algae named as chlorophyll, green algae, pheophyll, brown algae, rhodophyll, red algae, and their distinctive characters are also described there, right? We can find the difference in their pigments, difference in their reserve food, difference in the cell wall nature, difference in the flagellar number, arrangement and size, and also the habitat, right? So chemical composition of cell wall, that was listed there under chlorophyll. The cell wall is made of cellulose on inner side and pectose on outer side. Under pheophyll, the cell wall is made of cellulose and algin. And under rhodophyll, the cell wall is made of cellulose, pectin, and polysulfate esters. Structural organization of thallus, that was not at all described there, right? That means all the algal members are multicellular truly. Nature of stored material in the cell, that also was described there. Under chlorophyll, the reserve food is starch, which is coated over proteins called the pyrenoids present within the chloroplast. And under pheophyll, the brown algae, the reserve food is iodine-rich starch said to be the laminarin or mannitol. And under rhodophyll, the reserve food is somewhat complex, which is equivalent to that of glycogen, said to be the Floridian starch, right? And the type of pigments present in the cell that also were described there in that table. Under the chlorophyll, the pigments are chlorophyll A and B. For pheophyll, the pigments are chlorophyll A, C, and fucoxanthin. And for rhodophyll, pigments are chlorophyll A, chlorophyll D, and phycoerythrin. So the chemical composition of algal classes are quite different. The reserve food material is also different. At the same time, the pigments are also different. So which of these statements, which one is given with the most priority? That's the question. If you are asked to classify the various algae into distinct groups or classes, which of the following characters you should choose? Okay, so that should be the pigments present in them, right? The chlorophyll, the green algae, pheophyll, brown algae, rhodophyll, red algae are primarily different in their pigments, right? The major difference is there in their pigments. So the prior option meant for such a question is the types of pigments present in different algal classes. All right, that's it. And listen to the question number 9. Listen to the question, please. In a longitudinal section of a root, yeah, we can dissect the root or stem or any of the plant part in 2 planes, right? If it is a root, you can dissect either it's in this plane, that is said to be the cross section, the CS, or a transverse section, the TS, right? Or you can dissect the same root in lengthwise manner too, that is said to be the vertical section, VS, or longitudinal section, LS. Here the question is the question is meant for a dissection in its vertical manner. In a longitudinal section, the so-called LS, otherwise called the VS, right, of a root starting from the tip upward to the 4 zones occur in the following order. Options are here. You just listen to the options, please. Okay. Options are here. Option A, root cap at the base, cell division just above the root cap, cell enlargement and cell maturation, or else root cap, cell division, cell maturation followed by cell enlargement, cell division, cell enlargement, cell maturation and root cap, cell division, cell maturation, cell enlargement and root cap. Yeah, we are asked to detect the zones of root, yeah, that is upward from the tip upward. So at first you should mention the root part which is at the tip, that is said to be the root cap, right? Followed by the meristematic region, the zone of meristematic activity, meristematic cells meant for cell division, followed by enlargement, that means the elongation. Elongating cells are the enlarging cells, followed by the zone of maturation, the permanent part, right? So such an option is here in the option A itself. Root cap at the base, that means at the tip, followed by the cell division, that means the meristematic cells, followed by cell enlargement, that means the elongating cells, and thereafter only the cell maturation. The maturation zone will produce millions of unicellular hairs out, said to be the root hairs, right? So the right answer meant for this question number 9 is option A only. Okay, I hope you can follow me. All right, yes, listen to the question number 10. Study the following statements and choose the correct option. Yes. Of statement 1, buds are present in the axil of leaflets of the compound leaf. Pulvinous leaf base is present in some leguminous plants. Okay. In Elstonia, the petioles expand, become green and synthesize food. Is it? An opposite phyllotaxy is seen in Goa. Yes. Here the statements once again. Okay, you know, it's a compound leaf. It's a pinnately compound leaf like neem or curry leaf or so. It's a pinnately compound leaf. Okay, here it is a leaf base. Here comes the leaf base. It's actually the midrib called the rachis, and these are the veins, and the small leaf-like appendages are the leaflets. The first statement is stating that buds, the axillary buds, are present in the axil of leaflets, stating that the axillary buds are present at the axil of leaflets. No, we cannot see any of the axillary buds in the axil of leaflets. Instead, the axillary buds will be at the axil of the leaf base only. Here only you can see the axillary buds, not here, not at the axil of leaflets, but at the axil of a whole leaf you can find the axillary buds, right? So this statement is wrong, right? Axillary buds will not be at the axil of leaflets. Instead, the axillary bud will be present at the axil of the total leaf only. Okay. Statement 2. Pulvinous, if the leaf base is swelled, bulged, such a leaf base is said to be the pulvinous. Okay, such pulvinous leaf base is present in some of the leguminous members like a pea. Okay, the related member called the mimosa, the touch-me-not plant, etc. So that statement is pakka true. Pulvinous leaf base, the pulvinous leaf base is present in some leguminous plants. Fine. 3. Statement 3. In Elstonia, here comes the Elstonia, that is meant for the world phyllotaxy, more than 2 leaves are developing from a single node and they are arranged as a whorl, a layer around the stem, that is Elstonia. The petioles expand and become green and synthesize food? Not at all. That is stating the Australian acacia, right? The petioles expand and become synthesized food. The feature is shown by Australian acacia. That is not phyllo-clad, that is not cladoed, but it is phylloed, right? The phylloed is shown not by the Elstonia, but the phylloed is shown by Australian acacia. In Australian acacia plants, the leaf petiole expands and that attains a shape of a lamina and that synthesizes food. That is said to be phyllodes, which is not in Estonia, but it is present in the Australian acacia. So the third statement is wrong. Okay. The fourth one, opposite phyllodaxy. That means from a node, two leaves are developed, and the two leaves developing from the same node are opposite to each other is seen in Ga. Yes, Ga, Calotropis, etc. showing opposite phyllodaxy. So option, the statement 4 is also right. So of these 4 statements, 1 and 3 are wrong, but 2 and 4 are right. 1 and 4 are wrong. Okay. 1 and 3 are wrong. 1 and 3 are wrong. Okay. And 2 pulvinous. Okay. Leguminous plants, fine. And opposite phyllodaxy is seen in Gua. That also is right. So 1 and 3. 1 and 3 are incorrect. 2 and 4 are correct. That is option A itself, right? Option A. 2 and 4 are correct, but 1 and 2 are wrong. No, it's not so. 1 and 3 are incorrect. 1 and 3 are incorrect. 1 and 3 are incorrect. Though we cannot see 1 and 3 are wrong. Yeah. 2 and 4 are correct. Option A itself is the right answer. Yeah, fine. Moving on to the question number 11. 11th question is coming here. The flower of Fabaceae is Fabaceae. Yes, the family of pulses. Fabaceae. The flower of Fabaceae is, let us see, bisexual. Okay. Zygomorphic with bilateral symmetry. Okay. Gamosepalous condition, the sepals are united. Yes. Valvate or imbricate estivation also is right. Polypetalous, yes, with the papilionaceous corolla arranged as 1 plus 2 plus fused 2. One standard petal, the largest and outermost one. 2 lateral free wing petals and 2 fused keel petals, right. And under the option B, unisexual itself is wrong. So leave it. Option C, bisexual, okay. Zygomorphic, fine. Gamosepalous is okay. Imbricate, not only imbricate, sometimes they may be valvate too. And polypetalous, fine. So here comes a clarity issue. Bisexual, fine. Zygomorphic, fine. Persistent, no, the persistent calyx is a feature of Solanaceae, like tomato, potato, brinjal, etc. Valvate estivation, not only valvate, it can be imbricated too, right? So the best answer meant for the question number 11, the member of Fabaceae shows bisexual flower with both androecium and gynoecium together, where the androecium is made of a total of 10 stamens arranged as 9 together and 1 individually, said to be the diadelphous stamens, fine. And the gynoecium is monocarpellary with a superior ovary, so bisexual is the nature. Both the androecium and gynoecium are seen together. Zygomorphic, exactly, the flowers are bilateral in symmetry, said to be the zygomorphic nature. Gamosepalous, yes, calyx is made of 5 sepals, they are fused together called the gamosepalous, either in valvate or in imbricate estivation, right? Polypetalous, exactly, of the 5 petals, which is polypetalous in nature with vexillary estivation or papilionaceous corolla, where the largest outermost petal is the standard petal. 2 lateral ones are the wing petals, and the innermost, the smallest, fused ones are the keel petals. So option A only is the best answer meant for question number 11. Okay, question came from the chapter Morphology of Flowering Plants from the family description of Fabaceae. Yes, listen to the question number 12. An angiospermic plant has 24 chromosomes in microspore mother cells. Okay, the number of chromosomes in its endosperm will be 12, 24, 48 and 36. You know, the microspore mother cell, the microspore mother cells, they are said to be the pollen mother cells. Microspore mother cells themselves are the pollen mother cells, right? Abbreviated as PMC, fine. They will be diploid in nature. 2n. The 2n number is given here as 24. 2n is equal to 24. So, what will be the chromosome number in the endosperm? That is a question. The endosperm in angiosperm is developed by triple fusion, right? Triple fusion. The result of triple fusion is the PEN, where the central cell becomes the primary endosperm cell that will develop into a nutritive tissue called the endosperm for giving nourishment to the developing embryo, right? That triple fusion where both the polar nuclei fuses together. The first polar nucleus that will be haploid in nature that fuses with the second polar nucleus of central cell that also is haploid in nature. They both fuse together to form a secondary nucleus. That secondary nucleus will be diploid in nature. Okay, secondary nucleus is diploid, onto which a male gamete fuses together, thereby said to be the triple fusion that resulted in primary endosperm nucleus, that is 3n. Triploid in nature. So, the 2n pollen mother cell, the pollen mother cell, the microspore mother cell, the diploid chromosome number is 24. So, we have to count the triploid nature of its endosperm. So, that means its n is 12 and its 2n is 24. Thus, the 3n will be 36, right? The right answer is option D only. That is 36, right? Its haploid chromosome number is 12. Okay, in such a plant, the haploid basic set of chromosomes are 12. There's a diploid nature of mother cells are 24. 24 chromosomes will be there in the diploid cell. This is the triple endosperm should hold 36 chromosomes. Fine now? Okay. And question number 13. All the following statements regarding sieve tube elements are true except. True except. True except means we have to count the wrong one, right? We should count the wrong one. All other statements are true except one. The odd one is wrong. We have to identify the wrong statement. Chapter Anatomy of Flowering Plants, topic complex tissues and particularly phloem made of sieve tubes, companion cells, phloem parenchyma and phloem fibers. Options are here. Their end walls have perforated sieve plates, with the sieve pores, which become impregnated with lignin at maturity. They possess peripheral cytoplasm as well as a large vacuole at the center. C. Distinct proteinaceous inclusions, the P proteins, the so-called the phloem proteins, are seen evenly distributed throughout the lumen. And option D. Sieve tube elements are long, slender, tube-like structures arranged in longitudinal series. So, we have to count the incorrect one. We have to recognize the wrong statement from these four options. Okay. Sieve tube elements are long and slender, tube-like structures arranged longitudinally, right? They are arranged longitudinally. In longitudinal series, they were arranged. The option D is pakka true. Okay. Distinct proteinaceous inclusions, the so-called the phloem proteins, are seen evenly distributed throughout the lumen. Pakka true. That also is right. They possess a peripheral cytoplasm and a large central vacuole to carry the food in the form of sucrose. That also is fine. But their end walls have perforated sieve plate, that much is okay, which become impregnated with lignin. That is wrong. Sieve plates are not coated with the lignin, instead is coated with the callose. Okay, the sieve plates are coated with impregnated with the callose. So here the lignin is provided, that is wrong. So the wrong statement, the incorrect statement is option A only. Okay, that's it. And now we are moving on to our question number 14. Duramen, again chapter Anatomy of Flowering Plants, are from the secondary growth area. Duramen, otherwise said to be the heartwood, right? Duramen is the heartwood, the long durable wood. Duramen is present in inner region of secondary wood, part of sap wood, outer region of secondary wood, and region of pericycle. Yeah, duramen is otherwise said to be the heartwood. Duramen is the heartwood, the dark colored wood, right? The highly durable wood, the dense wood, right? And you know, sapwood is otherwise said to be the alburnum, right? Alburnum is the sapwood, alburnum, that is, which is quite less in density, and they are conductive in nature. That itself is the, alburnum itself is the outer region of secondary wood. And region of pericycle, pericycle is not at all a wood, right? Pericycle is not a wood. Wood is the xylem portion. So the duramen or the heartwood means the inner region of the secondary wood. That is the right answer. The inner region of the secondary wood, the inner region of the secondary xylem, occupied with the plant metabolites like tannins, oils, resins, gums, and syrup. There will be much in durability that can resist microbial action. That is said to be the heartwood or duramen. This inner region of the secondary wood, the early formed secondary xylem, that is the duramen, the heartwood. Okay, yes. Question number 15 is coming. In eubacteria or the true bacteria, a cellular component that resembles eukaryotic cell is options: nucleus, ribosomes, cell wall, and plasma membrane. Yes, the first option, nucleus, you know, nucleus is absent in bacteria, absent in all prokaryotes. So eubacteria are also devoid of nucleus. Regarding ribosome, the bacterial ribosome is 70S, made of smaller 30S and bigger 50S subunits. But in eukaryotes, the ribosome is 80S, made of smaller 40S and bigger 60S subunits. Cell wall, yeah, for bacteria there is a cell wall, but not all the eukaryotes are having cell wall, especially animals are lacking cell wall. So, the right answer meant for the question, in eubacteria, a cellular component that resembles eukaryotic cell is the plasma membrane alone. The plasma membrane, the lipid bilayer, right, made of two layers of lipids associated with the integral and peripheral proteins. The basic structure of that plasma membrane is unique in eukaryotes and prokaryotes. The right answer is option D from the chapter Cell, the Unit of Life. Okay, yes. Here comes question number 16. Ribosomes are found in options: cytoplasm and nucleus, Golgi complex and nucleus, mitochondria and bacterial cell, endoplasmic reticulum and Golgi complex. Yeah, assess yourself. Cytoplasm is holding ribosomes. The cytoplasmic ribosomes of prokaryotes are 70S, but the cytoplasmic ribosomes of eukaryotes are 80S. Nucleus is not at all holding any ribosome. Golgi complex is not holding ribosomes at all. Nucleus is also not holding ribosomes. Mitochondria shows ribosomes, they are 70S in nature. The prokaryotic ribosomes are present within the mitochondria and chloroplast, right? And the bacterial cell is also holding ribosomes, they are also 70S in nature. Endoplasmic reticulum, especially the rough endoplasmic reticulum, RER, is holding ribosome on its surface, they are 80S in nature. But Golgi complex is not holding ribosome. So from these options, both the components holding ribosomes, that is option C. Mitochondria is holding 70S ribosomes, and the bacterial cells are also holding 70S ribosomes. Thereby, the right answer is option C. Okay. Let us see the question number. The next question, question number 17. The G0 phase, the quiescent phase is options: phase after G2 phase, you just assess yourself what will be which phase will be after G2 phase, okay? Phase after M phase, okay, in which daughter cell enters new cell cycle, arrest of cell cycle on the onset of differentiation and the beginning of the cell division. Yeah, of these options, which will be the G0 phase? Phase after the M2 phase, G2 phase? No, because after the G2 phase there will be a phase called the M phase. Phase after the M phase, in which daughter cell enters new cycle. If the daughter cell enters a new cell cycle after the M phase, the G1 will be the arrest of cell cycle on the onset of differentiation. That will be the right option meant for G0 phase, the quiescent phase. The beginning of the cell division is marked by the karyokinases, the prophase of karyokinases, right? The beginning of the cell division is marked by prophase of the karyokinases, right? So the right answer is option C alone, arresting cell cycle, right? For they will not proliferate furthermore. The G0 phase cells will not proliferate furthermore. The eye cells, heart cells, etc. are in G0 phase. They are living, they are showing metabolism, but they will not proliferate furthermore, right? They will arrest the cell cycle on the onset of differentiation. That is the G0 phase. Yes, that's it. Yes, let us see the question number 18. Arrange the following events of meiosis in the correct sequence, like a terminalization, crossing over, synapses, and disjunction of genomes. Okay, you just see the things. Okay, the terminalization of chiasmatia that takes place in diakinesis stage of prophase 1, is it? Crossing over is marked in the Pachytene stage of prophase 1. Synapses, which takes place in the zygotine stage of prophase 1. These events are coming under the prophase 1 of meiosis, right? Disjunction of genomes, that is a feature of anaphase. During anaphase only, the disjunction, separation of genomes takes place, right? So of these events, which one will be at the earliest? Which one will be at the first? That is zygotine, right? Zygotine will be at first, right? And of these members, which one will be at the second? That is the Pachytene stage only, the crossing over stage only, right? Which one will be at the third position? That will be the diakinesis, right? From these options, that is diakinesis. Following the diakinesis, following the prophase 1 only, we can see the anaphase, right? So the right order, the correct order meant for these events will be zygotine, synapses, that should be 3 at first. 3 should be at first. Pachytene, that is a crossing over stage, the 2 will be at the second position, right? Followed by the diakinesis, where the terminalization of chiasmate is seen, that is 1 in number, and followed by the anaphase for disjunction, that will be at the 4th position, right? So the right answer meant for this question is option B only, right? Yeah, I hope it is okay for you, right? And listen to the question number 19. Here comes question number 19. Listen to the question. Wilting due to the plasmolysis, right? Wilting occurs when? Okay, wilting in plants occurs when? Chapter transport in plants, okay. Rate of transpiration is high than absorption. Rate of absorption is higher than transpiration. Excess root pressure is seen. High relative humidity in the air. So when you can see the wilting of a plant. Wilting of a plant means that plant is in a drooped down condition, right? Its leaves and all are in a drooped condition, wilted condition due to plasmolysis, due to the lack of water, right? So the wilting is seen when the water content is lacking in the plant body. When the water is lesser in the plant body, the cells become plasmolyzed and the plants will wilt, right? Such a condition is described here. Is it higher relative humidity in the air? No, right? If the relative humidity is higher in the atmosphere, the plant body will be in the most healthy condition. All the cells will be in the turgid nature. The leaf will be in the expanded position. So that's not at all possible. Excess root pressure is seen. If the root pressure is excess, that will push the water towards upside. Thus, the leaves will get more and more and more and water, and thus the leaves or the cells will be in the turgid stage, or also cause a turgidity, or the real the shape is exhibited. So that's not at all possible when the plant is wilted. Rate of absorption is higher than transpiration. Transpiration is low and absorption is high. Then also the cells will be in the turgid phase, and the plant body will be in a stretched condition. So rate of transpiration, water loss is high and absorption is less. Thus the plant will wilt. Right, due to the excessive transpiration, due to the excessive loss of water, the excessive loss of water in the form of vapor through the stomata, the plants will face a water potential decrease, okay, thereby the cells will become in a plasmalized stage and the plant parts become in a wilted condition. So that is the right answer. Wilting occurs when the transpiration rate is high and the absorption rate is low. Okay, yes. Here comes the question number 20. Munch hypothesis, again chapter transport in plants from the phloem translocation, right? Yeah, is based on, what is the principle of Munch hypothesis? Option A, translocation of food due to TP, turgor pressure gradient and imbibition force. Okay. Option B, translocation of food due to turgor pressure TP gradient. Option C, translocation of food due to imbibition force. D, translocation of food due to wall pressure. What is imbibition? A special type of diffusion, where the water is taken up by the dead solid colloids that leads to the increase in its volume. The process is said to be imbibition. That is not at all a living process. That is a property shown by the non-living particles, right? The non-living matter. So, translocation of food in the form of sucrose due to turgor pressure gradient, that much the statement is okay. And imbibition force, no. Imbibition force is not at all operated in the phloem. The Munch hypothesis is meant for phloem translocation. Okay? Phloem is mainly translocating organic food in the form of a disaccharide sucrose from a source towards the sink cell based on a turgor pressure gradient. Okay, the movement is downhill, the movement is downstream from a higher pressure region to a region of lower pressure, described by Munch, and thus said to be the Munch hypothesis. Second option, translocation of food, in the form of sucrose due to turgor pressure TP gradient. Yes, the TP of the source should be at the most and the TP, the turgor pressure of the sink, should be at the least condition. Thus, the food in the form of disaccharide sucrose will move from the source, a higher pressure region, to the sink, the lower pressure region. Option B is the right answer. Okay, so it's not at all based on imbibition, alright, it's not at all based on the wall pressure, but it is based on the turgor pressure. Okay, option B is the right answer for question number 20. Yes, here comes the question number 21. Minerals are absorbed by plants in option A, colloidal form, option B, ionic form, option C, precipitated form, and option D, both A and B, that means colloidal form and ionic form. You know, few of the non-mineral elements are obtained by plants from the atmosphere. They are carbon and oxygen. One another non-mineral element, that is hydrogen, which is taken up by the plants from the soil in the form of water, for that they split the water. The water oxidation takes place, called the hydrolysis or photolysis, to get the H plus ions, right? And rest of all the minerals, the remaining minerals, all are obtained or all are gained by the plants from the soil suspension, where all those minerals are occupied as charged ions. The minus charged anions or plus charged cation forms, and thereby most of the minerals are taken up by the plants from the soil in the form of ions. Okay, even though the concentration gradient is there, even though the concentration of ions are much more in the soil, even then the plants cannot obtain them in a passive way. That will not run in a diffusion, that will not run in simple diffusion manner. If the concentration gradient is there, the charged ions will show the facilitated diffusion. The facilitated diffusion. When the concentration of ions are more in the soil and less in the plants, thus the plant will uptake those minerals by using a process called the facilitated diffusion, where the energy is not required, ATP is not required, but that should be the carrier protein. And in most of the instances, the mineral concentration is much higher in the plant body and lower in the soil. Thus the plants will uptake those minerals in active form. Active transport should be there that will spend both energy, and that is with the help of carriers alone. So the minerals are absorbed by plants in ionic form, charged ion form, right? Yes, that's it. Here comes the question number 22 for you. Listen to me. Farmers in a particular region were concerned that premature yellowing, the premature yellowing of leaves, technically called the chlorosis, right? The premature yellowing called the chlorosis, a deficiency syndrome of a pulse, the Fabaceae member crop, might cause a decrease in the yield. Yes. Which treatment could be most beneficial to obtain maximum seed yield? We have to rectify the mineral deficiency that causes the chlorosis. Option A, frequent irrigation of the crop. Irrigation will provide water alone, right? That will not compensate any of the mineral deficiencies. So it's not true. Treatment of the plants with the cytokinin along with a small dose of nitrogenous fertilizer? No, nitrogenous fertilizer will not rectify the chlorosis, the premature chlorosis. A removal of all yellow leaves and spraying the remaining green leaves with a 2,4,5 trichlorophenoxyacetic acid, 2,4,5-T. That's a derivative of auxin that will not regain the chlorophyll content. Option D, application of iron, that is Fe, and magnesium, that is Mg, to promote synthesis of chlorophyll. That is the right answer, because iron is required for the synthesis of chlorophyll, and magnesium will maintain the ring structure of chlorophyll, right? Due to the deficiency of these iron and magnesium, the chlorosis will be present on plant body. The premature yellowing will be shown by the plants due to the deficiency of iron and magnesium. So by supplying those iron and magnesium to the plant body, the chlorosis can be reversed. Thereby the option D is the right answer. Okay? Yes. Question number 23. The C4 plants, the hatch and slack pathway, the plant with a Kranz anatomy, are photosynthetically more efficient than C3 plants, because the efficiency of C3 plants is less compared to the C4 plants. Why? That is a question. Option A, the carbon dioxide compensation point is more. Nothing so. Nothing so. Compensation point is nothing so. Such a discrimination is not so there in between the C3 and C4 plants. Carbon dioxide generated during photorespiration is trapped and recycled through PEP carboxylase? No. The photorespiration is not at all shown by the C4 plants. So there is no recycling of carbon from the photorespiration in C4 plants, because the photorespiration is shown by C3 plants only. The carbon dioxide efflux is not prevented. No, notice not at all. The stomata will open randomly, and due to based on the concentration gradient of the carbon may get in or get out from the plant body. They have more chloroplasts. That can be the right answer, because these C4 plants, the Hatch and Slack pathway plants, are showing Cran's anatomy. Right? In Cran's anatomy, you know, in Cran's anatomy, the central vascular bundles are surrounded by some specialized bundle sheath cells. Right? Around these vascular bundles, there will be bundle sheath cells. They are arranged compactly. The bundle sheath cells are arranged compactly without intercellular spaces, right? With more chloroplasts, right? And even beyond them, they have the normal mesophyll cells also, right? They can show the Cran's anatomy, the wreath-like anatomy, the other central vascular bundles are surrounded by bundle sheath cells and are even surrounded by the mesophyll cells, and thus the chloroplast will be in both mesophyll cells and in the bundle sheath cells. Thus the chloroplast amount is much more will be there in the C4 plants, and thus their efficiency is higher compared to the C3 plants. That's it. Okay. Yes. Listen to the question number 24. Consider the following statements, and followed by reading these statements, you have to choose whichever the statements are true and which one is wrong. Okay. The portion of the spectrum, the portion of the electromagnetic spectrum between 300 and 500 nanometers is also referred to as PAR, photosynthetically active radiation. Is it right or wrong? It's a blunder. It's a wrong statement, because the PAR, the photosynthetically active radiation, ranges from 400 to 700 nanometers, right? Yeah, that's it. The second statement is here. Magnesium, that is Mg, right? Magnesium, calcium, Ca, and chloride, that is the derivative of chlorine ion, play prominent role in the photolysis of water. Photolysis of water is otherwise said to be the hydrolysis, right? Yeah, hydrolysis in the presence of light is said to be the photolysis, right? Yes, that is the oxidation of water to emit protons, electrons and oxygen from the water, right? That is associated with the photosystem 2, the PS2 of thylakoid membrane, located towards the uppressed part of thylakoid membrane towards the inner surface close to the lumen, right? Where we can find a photolysing enzyme complex that is associated with the manganese, Mn, calcium and chlorine. Here it is stated as magnesium, so it's wrong. Actually, manganese should be the manganese, calcium and chlorine. They are the components of photolysing enzyme, not magnesium. Okay, so 2 is also wrong. And the third statement, in cyclic photophosphorylation, that is present in PS1, where the electrons are cycled within PS1, that is present on the stroma lamellae, the intergranule thylakoids, the stroma lamellae, where the electrons the path of electrons is circular, the electrons are cycled within PS1. Is it? ATP alone is produced, no NODPH is formed, right? And the photolysis, release of oxygen are absent, that is the cyclic photophosphorylation. In cyclic photophosphorylation, oxygen is not released, yeah, because of photolysis, the hydrolysis is lacking there. As there is no photolysis of water, yes, pakka. And NADPH is not produced, yes, because ATP alone is produced in cyclic photophosphorylation. So of these 3 statements, the first 2 statements are wrong, but the third one is true. Okay, yeah, the 1 and 2 are false, but the 3 is true. That is option B. Option B is the right answer meant for this question number 24. Okay, we are half the way, and here you can see the question number 25. Photosynthesis in C4 plants is relatively less limited by atmospheric carbon dioxide levels because, yes, you know, if the plant is a C3, So they are greatly dependent on the level of carbon dioxide. If only the carbon level is higher, the plants will show C3 cycle, the Calvin cycle. And if the carbon level is declined, the carbon dioxide level is lesser, thus the plant will show the C2 cycle, the so-called photorespiration, the phosphoglycolate pathway. It's a wasteful process. The C2 cycle is a wasteful process shown by the C3 plants when the level of carbon dioxide is less, oxygen, temperature and light are more, right? So photosynthesis in C4 plants is relatively less limited by atmospheric carbon dioxide because the C4 plants will show C4 cycle itself even if the carbon level is less or carbon dioxide level is high. Yeah, irrespective of the carbon level, the C4 plants will also always show C4 pathway where the photosynthetic pathway runs and its productivity will be higher, right? First option. 4 carbon acids are the initial carbon dioxide acceptors? Not at all. The initial carbon dioxide acceptor of C4 plants is PEP, the phosphoenol pyruvic acids or the phosphoenol pyruvates. They are 3 carbon compounds. The primary fixation of carbon dioxide is mediated by a PEP carboxylase. Fine, that's the right one. Pepcase, phosphoenol pyruvate carboxylase, that will always take carbon dioxide towards it, right? Effective pumping of carbon dioxide into bundle sheath cells? No, carbon dioxide is not at all pumped into the bundle sheath cells as such. Instead, the 4 carbon aspartic acid or malic acid were transported into the bundle sheath cell where those 4 carbon acids undergo decarboxylation to release carbon dioxide, right? So that's wrong. Rubisco, the rubulose 1,5-bisphosphate carboxylase oxygenase in C4 plants has higher affinity for carbon dioxide? Not at all, right? Rubisco is always showing affinity towards carbon dioxide. It's not the case with the C4 plants. The right answer meant for this question is option B. Is it? Yeah, the primary fixation of carbon dioxide in these plants are mediated by pepcase. It's not pepco. It's not phosphoenol pyruvate carboxylase oxygenase, but it is pepc. That means pep carboxylase, right? Pepco, right? Phosphoenol pyruvate carboxylase only, not oxygenase, right? If it was Rubisco, Rubisco will show both carboxylation and oxygenation. Thereby, it can uptake oxygen when the level of oxygen is higher in the atmosphere. That leads to the photorespiration, right? But in the C4 plants, it's the initial carbon fixation that takes place in the mesophyll cells, where the initial carboxylation enzyme is pepcase, phosphoenol pyruvate carboxylase, but not pepco, right? That's it. Option B is the right answer meant for question number 25. Yes, you can see the question number 26. Listen to the question. In which of the following reactions of glycolysis, yes, the question is from the chapter Respiration in Plants, does oxidation takes place? Yeah, it's just reverse process will be there in the photosynthesis in the Calvin cycle. That is the reduction process. Okay, here comes the oxidation. Yeah, while converting the glucose 6-phosphate to fructose 6-phosphate, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate or the diphosphoglycerate to 3-phosphoglycerate, phosphoenol pyruvate to the phosphoglycerate. Yeah, the oxidation reaction that will emit ATP and NADPH, right? That will emit NADH precisely. That oxidation step is conversion of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, that means 3-PGAL, 3-phosphoglyceraldehyde into 1,3-bisphosphoglyceric acid, BPGA. An intermediate stage will be there, that is the conversion of 3-phosphoglyceraldehyde into 3-phosphoglyceric acid. Okay, thereafter it will become 1,3-bisphosphoglyceric acid. That step releases what? NADH. Yeah, NADH. Okay, by the removal of protons Okay, the released protons will gain by NAD plus, and thus the NAD plus undergo reduction to form the NADH. That is the oxidation step here, where the 3PGAL undergo oxidation to release H plus, right? So the right answer meant for this question is option B. The conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceric acid that will emit protons. Okay, thus the glyceraldehyde 3-phosphate undergo oxidation. The released protons are taken up by NAD plus to form the NADH. That's it. Yes, question number 27 is awaiting for you. The rosette habit of cabbage. Yeah, cabbage is actually in a rosette in manner, right? That means the leaves are arranged in a closed manner because its internodes are very short. So the leaves are arranged in a very close condensed form. That is a cabbage. The rosette habit of cabbage can be changed. That can be changed, where the internodes can be elongated and the leaves will occupy in a spatially distinct spaces, and thus the photoperiodism will work, and that induce the flowering in cabbage. That is by the action of which hormone? Chapter Plant Growth and Development. IAA. IAA is the indole 3-acetic acid, a natural auxin. GA is the gibberellic acid. Okay. ABA is the abscisic acid, the growth retarding hormone. Ethafone is the derivative of ethylene. So, of these hormones, which one is meant for this process? This process is said to be bolting, right? The process is technically called the bolting, right? Bolting. The rosette-shaped plants are reversed by a process called the bolting by increasing the internodal axis length, which is done by gibberellic acid. Gibberellic acids can increase the internodal elongation, and thereby the gibberellic acids are applied in the sugar canes to increase the length of sugar cane to improve the sugar yield, and also applied in the grape inflorescence to increase the length of pedicels to spatially separate these grape fruits, right? The same gibberellic acid itself is applied in the cabbage to reverse its dwarfism, to reverse its rosette nature. So gibberellin can reverse dwarfism. Okay, gibberellin can rectify dwarfism by keeping the plants in a taller mode, right, by increasing axis length. Thus gibberellic acid is the right answer. Okay, here comes question number 28. There are 10 flowers in one individual plant of Crotalaria. In each microsporangium of every stamen of all the flowers, there are 30 microspore mother cells. How many pollen grains are formed from that plant? A very cunning question, a very twisting question. Here is stating the flower of Crotalaria. Okay, in each microsporangium, every stamen of all the flowers, there are 30 microspore mother cells. So there are 10 flowers. 10 flowers. Okay, 10 flowers are there. Okay, of an individual flower of Crotalaria. In each microsporangium of every stamen of all the flowers, there are 30 microspore mother cells. You know, anther. A typical anther shows 4 microsporangia. Okay, 4 microsporangia will be there in a typical anther. A typical anther shows 4 microsporangia, 1 in each thecus, 2 in each lobe, thus 4 in a total anther. So, and of these 4 microsporangium, each of the microsporangium is holding 30 microspore mother cells, right? So, 30 pollen mother cells. Pollen mother cells, the microspore mother cells, 30 microspore mother cells in each of the microsporangium. So, how many pollen grains are formed? That is the question. How many pollen grains are formed? Pollen number, we have to count out the pollen number. Once again, there are 10 flowers in one individual plant of Crotalaria. in each microsporangium of every stamen of all the flowers, there are 30 microspore mother cells. 30 microspore mother cells per microsporangium. Okay, in each microsporangium there are 30 pollen mother cells. Okay, so what will be the total number of pollen grains produced from such a microsporangium? Pollen mother cells are diploid and the pollen grains will be haploid, right? For that the meiosis takes place. So 30 into 4 pollen grains will be present in each microsporangium. Each microsporangium will hold 30 into 4 pollen grains. That means 120 pollen grains will be there in each microsporangium. Such 4 microsporangia will be there in an anther. So 120 into 4 what pollen grains will be there in each anther. So each anther will hold 120 into 4 pollen grains. That will be 480 pollen grains in an anther. Okay, and such 10 flowers will be there, right? 10 flowers are there. And you know, in Crotalaria, each flower will hold 10 stamens too. Okay, each flower will hold 10 stamens too, which is coming close to the Fabaceae. So there will be 120 pollen grains in each microsporangium. So there will be 4 microsporangia in an anther. So 120 into 4, 480 pollen grains will be there in an anther. Such 10 stamens, 10 anthers will be there in a flower. So 10 stamens of a flower. 10 stamens of a flower will hold 480 into 10. That means 4,800 pollen grains, right? And such 10 flowers are there. So of these 10 flowers, the 4,800 into 10. That means 48,000 pollen grains will be there. From each pollen mother cell, 4 pollen grains are formed. Okay, here 30 pollen mother cells are there per microsporangium. So from a single microsporangium, 120, 30 into 4, 120 pollen grains are created. Right? Such 4 microsporangia will be there in an anther. A typical anther will hold 4 microsporangia. So 120 into 4, 480 pollen grains are formed from an anther. Such 10 stamens will be there in a flower. So from a single flower of Crotalaria, 480 into 10, 4,800 pollens are formed. Such 10 flowers are there. So 4,800 into 10, that is the 48,000 pollen grains will be born from such a plant holding 10 flowers. Each flower is holding 10 stamens. Each stamen is holding the anther with 4 microsporangia. Each microsporangium is holding 30 pollen mother cells. Right? It's a nice question, a twisting question for you. Yeah. And now, question number 29 is awaiting. Pollen grains that were easily germinated on stigma are found to germinate in vitro when only 10 to 200 parts per million of boric acid is added. This suggests that, yes, a cunning question again, a critical question. Normally the pollens are easily germinated on the stigma, but in vitro condition within the laboratory, the pollen germination is possible only while provided with the 10 to 200 ppm of boric acid. What it indicates? Option A, boron accelerate protein synthesis in pollen grain. Boron has an abrasive effect on the exine. C, boric acid serves as a solvent for sporopollenin. And D, pollen wall is boron deficient and high levels of boron occur in the styles and stigma. While keeping the pollen grains as such in the laboratory, the pollen germination will not be seen. But by keeping the pollen grains on the stigma, pollen germination is done in an easy manner. Or it can be induced by providing 10 to 200 ppm of boric acid. So, it indicates what? It indicates by keeping the pollen grains as such, the pollen germination is prevented because the pollen wall is lacking the boron. And by providing the boric acid, the pollen germination is induced. Naturally, it is provided by the stigma. So, option D will be the right one. Pollen wall is boron deficient. So, as such, they will not germinate as such, right? And high levels of boron occur in the styles and stigma. Thus, on the stigma, the pollen germination will take place in an easy way. That is the right answer. Pollen wall is boron deficient and high levels of boron occur in the style and stigma. Boron is a must for pollen germination, pollen tube elongation, cell enlargement, etc. Okay. All fine. Here comes the question number 30. The eyes of the potato tuber are flower buds, shoot buds, axillary buds, and root buds. You know, the edible part of the potato, the so-called potato tuber, is the stem tuber, right? Is the stem tuber, is the main stem, all right. And you can see several small depressions on the stem tuber. Those depressions are said to be the eyes. Eyes are represented by the nodes. Okay. By keeping these potatoes in the kitchen cabin, by keeping them in the kitchen shelf, they will sprout out with the influence of ethylene. Ethylene will induce the sprouting of potato. The gaseous hormone ethylene, C2H4, will induce the sprouting of potato. And by keeping them in kitchen shelf, they will sprout out. So, asking regarding these sprouts, right? Asking regarding the question is posing. Question is posed towards these sprouts coming from the nodes of the potato tuber. The eyes, the nodes of portion of the potato tuber represents actually the axillary buds. The eyes, actually they are the nodes that will produce the axillary buds, right? The option node is not at all here, so we are giving the second prior option, that is the axillary bud. The eyes, the small depressions on the potato tuber that will produce the axillary buds, okay? And the sprouting of potato is done by the hormone called ethylene. All fine. Question number 31 is here. An interesting modification of flower shape for insect pollination occurs in some orchids, in which a male insect mistakes the pattern on the orchid flower for the female of his species and tries to copulate with it, thereby pollinating the flower. This phenomenon is called, we are asked with the question, tries to copulate with it. That is the question. Options: pseudo-parthenocarpy, mimicry, pseudo-pollination, and pseudo-copulation. If the question was regarding the imitation of a flower, the flower is imitating one of the insects, thus the option mimicry will be the apt one. But here, the question is posing towards such a flower, such an insect is trying to copulate the flower. So the phenomenon, the process of copulation, that is the question, that is called the pseudo-copulation. The pseudo-copulation shown by certain orchids through its mimicry by imitating certain insects. Parthenocarpy is the process of forming fruits without fertilization, producing seedless fruits. Mimicry is the imitation of a flower. A flower is imitating one of the insects, that is mimicry. But here the copulation process is asked for, that is called the pseudo-copulation. Okay, pseudo-pollination, there is no concept like this. Okay. Question number 32. Gestation period is the duration between, actually a zoology question, but we can answer is from the chapter Reproduction in Organisms. Gestation period is the duration between ovulation and fertilization? No. Maturation of egg and ovulation? No. It is the period between fertilization and parturition, right? The period between the fertilization and parturition is said to be the gestation period. Option C is the right one. It's a straight question from the NCERT from human reproduction, right? Which one of the following is not a biofertilizer? Options are provided here: Rhizobium, Nostoc, Mycorrhiza, Agrobacterium. Yeah, you just assess yourself and try to crack the right answer. Rhizobium is a symbiotic nitrogen-fixing bacterium associated with the roots of leguminous plants. Thus, the rhizobium is associated with biofertilization, biofertilizer providing nature, providing nitrogen to the leguminous plants. Nostoc is associated with the coralloid roots of cycas, as well as the thallus of Anthoceros, etc. We can see the specialized cells called heterosysmen for nitrogen fixation. They also can provide fertilizer, the nitrogen, to the host plant. Mycorrhiza is a symbiotic association of fungal mycelium with the roots of higher plants like pinus, where the surface area of absorption can be increased. The fungal partner can uptake water and minerals from the soil. Okay, but the Agrobacterium, the natural Agrobacterium is a pathogen, right? The Agrobacterium is a pathogen holding Ti plasmid, the tumor-inducing plasmid. That is a pathogen. A tumor-causing pathogen is the Agrobacterium tumefaciens, a common soil bacterium. By disarming them, by genetically modifying them, they can be used as vectors in biotechnology, right? So the natural Agrobacterium tumefaciens is a pathogen that will cause disease, the tumor disease in roots of dicot plants. Okay. So that is the right answer. Agrobacterium is not at all a biofertilizer. Instead, rhizobium, nostoc and mycorrhiza all are biofertilizers. Yes, fine. Question number 34 is here. Can you follow me here? Can you recollect the concepts from the corresponding chapters? I hope it's okay for you, right? Question number 34 is awaiting. Which one of the following pesticides is banned nowadays? A non-biodegradable pesticide is banned nowadays. Which one it is? DDT, eldrin, aldrin and toxaphene. Yeah, the question is from the chapter environmental issues, right? Yeah, environmental issues is holding a concept like biomagnification. The biological magnification is called the biomagnification. Yeah, the magnification, accumulation of certain things in living bodies is called the biomagnification, the biological magnification, where the non-biodegradable pollutants will accumulate in the highest trophic levels of a food chain. Okay, that can be the accumulation of mercury, the accumulation of DDT, etc. So DDT is the answer. DDT is the answer. DDT is a non-biodegradable pollutant that is accumulated in the highest trophic levels, accumulated in phytoplanktons, then in zooplanktons, much more magnified in the small fishes and again in the large fishes, right? That is DDT, and thereby the DDT is banned nowadays. Okay. Question number 35 is here. Breeding of crops. The artificial hybridization of crops with the higher levels of minerals, yeah, improving the food quality. Higher levels of minerals, vitamins and proteins is called. That means the plant breeding for improved food quality. The plant breeding meant for improved food quality. Somatic hybridization? No. Somatic hybridization is the fusion of protoplast from somatic cells. That is potato plus tomato, right? Biofortification? That is the right answer. Biomagnification? No. Biomagnification is the accumulation of non-biodegradable pollutants in the highest trophic levels of food chains. And micropropagation? No. Micropropagation is the tissue culture itself. During tissue culture, thousands of plantlets can be released from few of the explant cells, and thus the tissue culture is otherwise said to be the micropropagation. So the right answer is biofortification, is not biomagnification. Okay, biomagnification is the accumulation of toxicants, the non-biodegradable pollutants in the highest trophic levels of food chain. But here it is the biofortification, the plant breeding for improved food quality, improving protein content and quality, vitamin content and quality, mineral nutrient contents, oil or fat contents and quality called the biofortification. Examples are golden rice with vitamin A, Atlas 66 wheat with the proteins. Okay, the biofortified rice with the 5 times improved iodine content. The maize released in 2000 with double the twice amount of amino acids like lysine and tryptophan. And for Indians, the biofortification is done by Indian Agricultural Research Institute IARI located at New Delhi. They were released vitamin A enriched carrot, spinach and pumpkin, vitamin C enriched bitter gourd, badwa, mustard and tomato, iron and calcium enriched spinach and badwa, and protein enriched beans like broad, lab, French and garden peas, right? That is biofortification. The biofortification is the right answer meant for the question number 35. Okay, here comes the question number 36. Desired improved varieties of economically useful crops, the food crops, are raised by migration. Migration is a temporary two-way movement of organisms from a stressful habitat to a more hospitable area, and they will get back when the original conditions become favorable. Biofertilizer. Biofertilizer is the fertilizer obtained from living bodies, right? Hybridization, that is artificial hybridization. Germplasm collection, evaluation and selection of parents, cross hybridization among selected parents through emasculation, bagging, artificial pollination, rebagging, collection of seeds from the bag to form F1 generation, right? Yeah, testing and release of the superior recombinants and commercialization. So the right answer is hybridization. That is a method inculcated to produce economically useful varieties and improved crops, right? Natural selection is a natural phenomenon, right? A natural force, a nature's force applicable to the diverse organisms to choose the best ones and to eliminate the worst ones, right? That is a natural selection that leads to the survival of fittest. So here the right answer is option C only. Okay. Question number 37 is coming. Maximum application of animal cell culture technology, like any of the cells, HeLa cells, right? Or animal cells, anything, today is in the production of vaccines, edible proteins, insulin, and interferons. Interferons are present in insects, right? Yeah, so insects only. Insulin is obtained from E. coli, right? The American company Eli Lilly in 1983 patented the human insulin production. The human production, that is by using E. coli, the genetically modified E. coli. Edible proteins are obtained from single cell proteins, right? SCP, like spirulina is a monorin, okay, chlorella, yeast, mushrooms, Methylophilus, Methylotropus, etc. So the right answer is meant for vaccines. Okay, the maximum application of animal cell culture technology today is in the production of vaccines. Vaccine production. For the vaccine production, the animal cultures are maintained in the laboratory mostly. 38. Polyethylene glycol, otherwise said to be the PEG. Polyethylene glycol, the PEG. The polyethylene glycol method is used for what? The gene transfer without a vector, biodiesel production, seedless fruit production, and energy production from sewage. Yeah, it's not the energy production from sewage. From sewage, the biogas can be produced using the methanogen bacterium. Seedless production is called the parthenocarpy. For the parthenocarpy, hormones like auxins and gibberellins are applied. And biodiesel production, for that the plant Jatropha. The Jatropha plant is used for the biodiesel production. And thus PEG is used for gene transfer without a vector in plant cells, right? In plant cells, the external gene can be penetrated into the plant cells using polyethylene glycol. So PG is used for the gene transfer in plant cells without a vector. Okay. 39th question. Precipitates of purified DNA after the addition of chilled ethanol can be seen as a collection of fine threads in the suspension. Chapter Biotechnology Principles and Processes, DNA Isolation. Is it? Yeah. By the removal of cell walls, the protoplast can be obtained. By grinding those protoplasts using mortar and pestle, a mixture paste can be obtained. From the mixture, the RNA can be removed by ribonucleases, protein with the proteases, lipids with the lipases, carbohydrates with the amylases, at last the DNA alone will be remained at the bottom of the test tube. That can be precipitated up using the chilled ethanol, right, as a precipitating agent. This process is referred to as what? DNA transformation? No. Transformation is the process of transferring a gene from one organism to the other organism. DNA ligation? No. DNA ligation is the linking of DNA fragments using DNA ligase enzyme. DNA spooling, that is the right answer. Spooling, the precipitation of DNA and collecting it from the chilled ethanol is called the pooling, right? DNA duplication? Duplication is no, not at all, because duplication is the replication itself, right? Duplication of DNA is the replication of DNA itself that naturally takes place in the S phase of cell cycle, or artificially can be done using PCR, the polymerase chain reaction, using a thermostable polymerase enzyme called the Taq polymerase. So the precipitation of DNA and its collection is called the DNA spooling. Okay. Alright, the 40th question. Genetic engineering has been successfully used for producing what? Transgenic mice for testing safety of polio vaccine before use in humans as part of downstream processing. Transgenic models for studying new treatments for certain cardiac diseases. Transgenic cow Rosie, which produces high-fat milk for making ghee. Animals like bulls for farm work as they have superpower. No, it's not so. Animals like bulls for farm work as they have superpower, for that the genetic engineering is not yet conducted. The transgenic cow Rosie is enriched with the human protein milk, right? Human protein enriched milk, right? The Rosie is producing human protein enriched milk, alpha lactalbumin, not the fat, high fat milk, right? Yeah. And the transgenic models for studying cardiac diseases? No. But the transgenesis, the genetic modification, the genetic engineering nowadays is successfully used for transgenic mice production for testing safety of polio vaccine before using them in humans as part of downstream processing. This option A is the right answer. Okay, of the transgenic animals, there are transgenic animals like transgenic cow, transgenic rabbits, transgenic pig, fishes, etc. But more than 95% of transgenic animals are mice, because the chromosome number of mice is somewhat similar to that of humans. Humans are holding 46 chromosomes, but the mice is holding 42, right? And you know, the mice, the more than 95% of the transgenic animals are mice, used for genetic studies, like the application of the downstream processing of safety studies of vaccine, like polio vaccine. Yeah, we are hitting the last 10 of our questions. Question number 41. Given figures represent the steps involved in polymerase chain reaction, that is PCR. Identify the steps A, B, C and D and select the correct option. Yeah, A is here. Here only you can see the A. Okay. And here comes the B. And here only is the C. And the D is a whole process, right? Okay. So from above you can see these things. Okay. yeah, you have to identify the steps denoted as A, B, C and D. A is the separation of both the strands of DNA by breaking the hydrogen bonds. So the A is the DNA denaturation. A is the denaturation that takes place in a very high temperature, that is 94 up to 98 degrees Celsius. Okay, the separation of DNA fragments that takes place in the DNA denaturation process, right? That is done in 94, 95, 96, 97 or up to 98 degrees Celsius. And B is represented here, you know, where the templates are added with the primers. The oligonucleotide sequences are said to be the primers. The primers are added. The primers are added onto the templates, onto the 3' end of templates. So the process is called the primer annealing. That primer annealing that takes place in between 40 to 60 degrees Celsius. Okay, and you know the C is marked here where the DNTPs are involved. Magnesium is facilitating the process where the new strand synthesis takes place. The complementary strand of template is produced. The process is called the primer extension or the strand extension that takes place in 72 degrees Celsius. And you know, D is represented here. The letter D is given here. That is the repetition of the process again and again and again, right? In a chain manner, in a chain reaction manner, in an unending manner, to double the DNA content in each cycle to form a maximum of billion DNA copies. So such a statement, we have to opt such a statement. A denaturation at 94.96 degrees Celsius? No, that is 94 to 96 degrees Celsius. A denaturation at 94 to 96 degrees Celsius? Fine, that is okay. Okay. B is the annealing at 40 to 60 degrees Celsius? Again okay, right? C, extension through Taq polymerase at 72 degrees Celsius? Fine, again it's true. And D, repetition of the denaturation and polymerization activity again and again? Exactly. This B is the right answer meant for this picture. Comes from chapter Biotechnology Principles and Processes, topic PCR, right? Option B is the right answer. Okay. Have a look to the question number 42. A technique which involves deliberate manipulation of genes within or between species. Manipulation of genes within or between species. Gene therapy, hybridoma technology, tissue culture, genetic engineering. Gene therapy is a process of genetic engineering to cure, to solve any of the genetic disorders. Okay? Yeah. The question is not giving emphasis to disease solving process, right? So it's not gene therapy. Hybridoma technology is familiar in oncology, in cancer studies. Hybridoma is a hybrid cell formed by the antigen created lymphocyte binded on to the tumor cell. If the antibody created lymphocyte is linked with the tumor cell, such a hybrid cell is called the hybridoma. That is in oncology. That's not here. A technique in which involves deliberate manipulation of genes within or between species. It's not giving any of the connection with the tumor cell or lymphocyte or so it's not hybridoma. Tissue culture, no, because tissue culture is one of the artificial asexual reproduction, in vitro vegetative propagation in plants using the totipotency as a principle, right? Producing thousands of soma clone plants from a parent. And thus the right answer is genetic engineering. Genetic engineering is the technique in which the manipulation of genes within or between the species is done. That is genetic engineering only. Option D is the right one. Yes. Question number 43. Which one is the edaphic factor? You know, edaphic. edaphic factor in the biosphere? Light, temperature, water and soil. So straight question, edaphic factor means soil. The soil related factor, that means the size of grains of soil, the water holding capacity of soil, the fertilizers in the soil, even the microbes in the soil, anything connected with the soil is said to be the edaphic. The soil related factors are said to be the edaphic factors. Okay? Question number 44 is here. The assemblage of all the populations of different species, okay, all the populations of the different species, that means different species, different populations. Yeah, one of the population is represented like this, yet another one as this, or else another population is represented as this. So a group of different populations, a group of different species are here. Okay, the group of same species is said to be the population. A group of similar species is said to be the population. Then the assemblage of all the population of different species that function as an integrated unit through co-evolved metabolic transformation in a specific area. The different populations living together in area is called biome? No, biome is special type of ecosystem, the large ecosystem with an over-dominating population called biome. The forest can be taken as an ecosystem or can be treated as a biome, right? Biotic community, that is our right answer. Okay, group of same species called the population, and the group of different populations called the community, the biotic community. Population is a group of similar species. Ecosystem is the community and it's a non-living components like light, temperature, water, etc. So the group of different populations is called the community, the biotic community. Okay. Question number 45. In short day plants, the plant is short day, by giving a pie diagram, if it is a short day plant, then the most area of a day is dark, right? In a short day plant, the short day plants will flower only when getting a short span of light, right? The night is prolonged and the light period is very less, right? Alright, so in a short day plant, in short day plants, flowering is induced by photo period less than 12 hours. There is no such a criterion as 12 hours day or 12 hours night. There is nothing so, right? Instead we are using the critical day length and the critical night length only. Photo period below a critical length and long night. A long night and the day length which is lesser than a critical day length. That's the right one, right? Long night only is not at all applicable towards the short day plants because the short day span also should be mentioned. Short photo period and interrupted a long night. If the night is interrupted, the plant will not flower. So the right answer is option B alone. The photo period should be below a critical length and the night should be very long and the night should not be interrupted. That's option B is the right answer. Okay. And now here is our question number 46. Loading of phloem is related to chapter again, transport in plants, phloem loading, the Munch hypothesis, right? The loading of phloem is related to what process? Increase of sugar in the phloem, okay? Elongation of phloem cell, elongation of the phloem cell, separation of the phloem parenchyma, and strengthening of phloem fibers. What is a phloem loading? Phloem loading is the two-step transport of sucrose, right? The sucrose is transported at first from the source cell to the companion cell of phloem and from the companion cell to the sieve tube of the phloem. Okay, the two-step active transport of sucrose at first from the source cell to the companion cell and then to the sieve tube associated with the pumping out of protons is called the phloem loading. Thus, the phloem loading is associated with the increase of sugar in the phloem, increase of sucrose in the phloem, increasing the food of phloem. That is phloem loading. Okay. Yes. 47. A cell divides every 1 minute. Okay. A single cell which is divided into two daughter cells using a single minute. Okay. Using a single minute, a cell is divided. A cell divides every 1 minute. At this rate of division, it can fill a 100 ml of beaker in 1 hour. Yeah, okay. The 100 ml beaker. Okay. The 100 ml beaker. The 100 ml can be divided into the first 50 ml and the second 50 ml. Okay. Yeah, all right. Okay. But yeah, in order to fill such a 100 ml beaker, the time required is just 1 hour. For this process, a single hour is required. That means 60 minutes, right? 60 minutes is required. Okay. Then question, how much time does it take to fill a 50 ml beaker? So, in such a rate, how much time it will required to fill a 50 ml beaker? That is the question. Options 30 minutes, 60 minutes, 59 minutes and 32 minutes. The question, the frame is given here. Yeah, the bacterial cell, the cell divides every 1 minute. For a single division, a single minute alone is required. So, in order to fill the 100 ml beaker, 60 minutes are required, right? Okay. That means by reducing a single minute from this, at the 59th minute, the beaker is half filled, right? That means the beaker is filled with 50 ml, right? Is it? Yeah. That means using a single minute, the 50 ml can be converted into 100 ml. Because for the doubling of the cell, a single minute alone is required. Just to double a cell, a single minute is required. So, if the 50 ml is there in the 49th minute, then by using the single next minute, the 16th minute, the volume will be doubled. Right? That means the time required to fill the 50 ml beaker is 59 minutes. Okay. By using the just next 1 minute, the 60th minute, the 50 ml will be doubled into 100 ml. Okay. Thereby the option C is the right answer, 59 minutes. Fine. Question number 48 is waiting for you. Select the wrong statement. A. When tripalmitin is used as a substrate in respiration, the RQ is 0.7. Is it true or false? Tripalmitin is a fatty acid where 1 NO2 carbon dioxide will be emitted. For that, 145 oxygen should be consumed. Thus, its RQ value will be 0.7. So, option A is true. Right? 2. B. The intermediate compound which links glycolysis with the Krebs cycle is malic acid. Malic acid is a 4-carbon organic acid, right? Yeah, the intermediate compound which links glycolysis with the Krebs cycle is malic acid. That is a pakka wrong, because the linking is done by the glycolysis is linked with the Krebs cycle using acetyl coenzyme a 2-carbon substance. So, second statement is wrong. C. One glucose molecule yields a net gain of 36 ATP molecules during aerobic respiration. Yeah, either 36 or 38 ATP can be produced based upon the conditions, right? So, based on our NCERT statements, yeah, especially the old NCERT, this statement is pakka okay. And D. One glucose molecule yields a net gain of 2 ATP molecule during fermentation. Yes, Fermentation is the anaerobic respiration, the partial oxidation of glucose that will release just less than 7% of energy from a glucose, that will be 2 ATP alone. So the wrong statement is option B only, because the linking molecule between glycolysis and Krebs cycle is not malic acid, instead it is acetyl coenzyme A. Okay. Alright, here comes our second last question from the chapter Plant Kingdom. Which of the following correctly represents the type of life cycle patterns from the options given? Yeah, the three pictures are given here. In the first picture, you know, yeah, the sporophyte is represented, zygote is represented here, that is diploid. The developed sporophyte is represented here, that again is diploid. During gametogenesis, the gametes formed alone are haploid. Under zygamy, the 2n condition will be restored. In such a life cycle, the predominant stages all are represented by the sporophyte, the diploid stage. The gametophyte is primitive. Thus, this life cycle is diplontic life cycle, shown by an alga called the fucus, and all the seed-bearing plants like gymnosperms and angiosperms are also showing diplontic life cycle. And here you know the gametophyte is haploid here, gametogenesis, fine. Under zygamy, the zygote alone will be diploid in nature, and by meiosis the spores, the haploid is again restored. So the sporophyte is represented only by the zygote. The remaining stages all are represented by the haploid gametophytic stage. Thus it is haplontic life cycle, shown by the colonial alga like volvox, filamentous alga like spirogyra, and a protistan like chlamydomonas. And here you know gametophytic stage is prominent, gametogenesis, and zygamy restores the zygote, by a sporophyte, but meiosis, the haploid spores are again restored. Both the haploid and diploid stages are equally prominent. Thus the third picture is showing haplodiplontic life cycle, an intermediate life cycle in between the haplontic and diplontic, shown by all the bryophytes, all the pteridophytes, ectocarpus, polysiphonia, and the kelps. So the first picture diplontic, second one is haplontic, and the third one is haplodiplontic. Okay, diplontic, haplontic, haplodiplontic. Option D is the right answer. Okay, and here comes our last question, the question number 50. Meristematic tissue are, question from chapter Anatomy of Flowering Plants from the topic meristem. Okay, meristematic tissues are premature having ability of division, mature does not have ability to division, premature not having ability to division, complex differentiating in xylem, phloem and cambium. Okay, complex differentiating in xylem, phloem and cambium, that's not meristem. Cambium is meristem, fine, alright, but xylem and phloem are not at all meristem, they are complex permanent tissues. Premature, fine. Okay, having premature, not having, so wrong. Okay, mature, no, wrong. Premature, okay, having ability of division, fine. Thus the right answer meant for meristematic tissues is option A. They are premature and they are having the ability of cell division. Okay, meristos means divided. Meristematic tissues means the tissues they are capable of division. They are living with a nucleus and cytoplasm called the protoplasm. They have a thin primary cellulosic cell wall with numerous plasmodesmata. Such cells are arranged compactly without intercellular spaces. Their vacuoles are either absent or very small. All those are the features of meristematic tissues. Yes, that much was our exam questions, 50 questions, alright? And I think we moved on through all those 19 chapters of botany, all the 11 chapters from plus 1 and 8 chapters from plus 2, right? I hope it's beneficial for you as a revision point of view. right? Yes, thank you so much for watching and listening me, and definitely I appreciate your patience and your time too, right? And thus it's wishes from our side, from the team of Cubeta Mercury, to each and every one of you for a bright future, especially for a better luck in your upcoming NEET exams. Thank you so much. Thanks a lot.